Welcome to the Hinterleitner lab. We are interested in the fascinating yet complex events happening in our intestines. We are trying to understand the crosstalk between intestinal microbes and the host mucosal immune system. When things go wrong in the intestine, for example a viral infection, the fine-tuned gut homeostasis gets out of balance, resulting in inflammation and disease. We currently have two main projects ongoing in the lab where you can contribute to, but own ideas are always welcome. Let me introduce the projects. In Project 1, we ask the question, how can we use gut microbes to protect from immune-mediated food sensitivities that include food allergies and celiac disease? Immune-mediated food sensitivities are increasing worldwide. It is estimated that 1 in 20 Americans suffer from food sensitivities. Elimination of the causative foods from the diet is the current standard and only care, but always bears the risk of cross-contamination with the offending food, which can significantly impact health in adults and especially children. The mechanisms underlying the increased incidence of food sensitivities remain unclear. However, several studies demonstrated that gut microbes, along with other host predisposing factors, dictate the development of immune-mediated food sensitivities. Gut microbes are increasingly considered as key modulators of mucosal immune responses to dietary antigens that protect from immune-mediated food sensitivities. To target microbe-induced immune pathways for intervention, it is crucial to understand the functional mechanisms of how microbes modulate immune responses to food antigens. Over the last years, there was a significant progress in our understanding of how commensal gut bacteria and enteric viral infections modulate mucosal immune responses in the context of food sensitivities. We recently showed that certain viral infections lead to loss of oral tolerance to dietary gluten and the development of celiac disease. The mammalian gut microbiome encompasses bacteria, fungi, archaea, and protists that contribute to gut health. We ask the question whether certain gut microbes can prevent inflammatory immune responses to dietary antigens in the context of celiac disease and food allergies, and, if so, what are the microbial-induced mechanisms of this beneficial immunomodulatory phenomenon? In Project 2, we ask the question, what are the regulators of type 2 immune responses in parasitic infections? Certain parasite infections that include helminths and protists can cause disease and are strongly associated with protective type 2 immune-mediated intestinal inflammation observed across species, including mice and humans, characterized by ILC2s, eosinophil and mast cell recruitment, and tissue injury. Type 2 immune responses are initiated by parasite-sensing interleukin-25 producing epithelial tuft cells, resulting in the activation of interleukin-13 producing ILC2s that in turn signal back on epithelial cells. This feed-forward IL-25 ILC2 IL-13 circuit amplifies type 2 immune responses and initiates massive tissue remodeling, including tuft and goblet cell hyperplasia and mucus production, resulting in the containment of intestinal parasites. Despite the recent progress in our understanding in immune tissue crosstalk in type 2 mediated inflammation, we lack a complete understanding of the detailed processes underlying parasitic-induced type 2 immune responses in the gut. This limits effective treatment options for enteric parasitic infections. To characterize type 2 mediated inflammation in the intestine in greater detail, we screened for novel players implicated in intestinal type 2 inflammation. We identified a set of highly homologous genes that were significantly upregulated in the intestine of mice upon type 2 inducing parasitic infections in vivo and in small intestinal organoid cultures upon IL-13 stimulating conditions in vitro. The goal of this project is to study the role of newly identified type 2 inducible genes in the protective type 2 immunity to protists and helminths, and their function in intestinal epithelial cells.